This is a diagram of two neurons. A neuron is a cell which transmits signals. Most of the neuron's organelles are located here in the cell body. The neuron receives the signal through the dendrites, which are these branches, and then the signal travels into the axon hillock where a new signal is created. And then this signal travels down the axon. The axon then branches into synaptic terminals, which will transmit the signal through a junction called the synapse. The synaptic terminals have vacuoles containing neurotransmitters, which transfer the signal, and then these neurotransmitters are released into the intracellular space, and then they travel through the postsynaptic membrane into the postsynaptic cell. This cell is the presynaptic cell because it makes the signal. This is the postsynaptic cell because it receives the signal. The transmission of a signal across a neuron can be described by the depolarization and hyperpolarization of a membrane potential. First, we will discuss how the signal is created in the axon hillock.
A membrane potential is created by a voltage across the plasma membrane. The voltage is the charge difference, or the unequal distribution of ions between the interior and the outside of the cell. For a resting neuron, or one that is not sending a signal, the membrane potential is called a resting potential. And when a neuron receives a stimulus, the membrane potential changes. These changes are known as action potentials. When creating a resting potential, sodium and potassium ions play an essential role. They both have a concentration gradient across the plasma membrane of the neuronal cell, and it's maintained by the sodium-potassium pump. The pump actively transports sodium out of the cell and potassium into the cell, but the pump acts slowly and causes the membrane potential to be very small. That's why ion channels are important, because they allow ions to diffuse back and forth across the membrane, carrying with them units of electrical charge. Ions moving rapidly through the channels generates a membrane potential. These ion channels also have selective permeability. For establishing a resting potential, it is critical for potassium channels to always stay open for the diffusion of potassium ions. With many potassium channels but very few sodium channels, a negative net charge builds up inside the cell, which is a major source of the membrane potential.
This graph shows graded depolarizations produced by two stimuli that increase membrane permeability to sodium ions. The larger stimulus produces a larger depolarization. The reduction in the magnitude of the membrane potential is called a depolarization. This graph shows graded hyperpolarizations produced by two stimuli that increase membrane permeability to potassium ions. The larger stimulus produces a larger hyperpolarization. The increase in the magnitude of the membrane potential is called a hyperpolarization. Action potentials are the nerve impulses or signals that carry information along an axon. At the resting state, the gated sodium and potassium channels are closed, and ungated channels maintain the resting potential. When a stimulus depolarizes the membrane, some gated sodium channels open, allowing more sodium to diffuse into the cell. If the depolarization reaches the threshold, it triggers an action potential. In the rising phase of the action potential, depolarization opens most sodium channels while the potassium channels remain closed. Sodium influx makes the inside of the membrane positive with respect to the outside. In the falling phase of the action potential, most sodium channels become inactivated, blocking sodium inflow. Most potassium channels open, permitting potassium outflow, which makes the inside of the cell negative again. In the final phase of an action potential, which is known as the undershoot, the sodium channels close, but some potassium channels are still open. As these potassium channels close and the sodium channels become unblocked, the membrane returns to its resting state.
Now we'll take a look at how this action potential travels down the axon.
An action potential functions as a long-distance signal by regenerating itself as it travels from the cell body to the synaptic terminals. At the site where an action potential is initiated, Na plus inflow, when it rises, creates an electrical current that depolarizes the neighboring region of the axon membrane. The depolarization in the neighboring region is large enough to reach the threshold, causing the action potential to begin there. This process happens again and again as the action potential travels the extent of the axon. At each position along the axon, the process is the same. so that the shape and size of the action potential remain constant. Immediately behind the traveling area of depolarization due to Na plus inflow is an area of repolarization due to K plus outflow. In the repolarized area, the sodium channels remain inactivated. Therefore, the inward current that depolarizes the axon membrane ahead of the action potential also cannot produce another action potential behind it. This prevents action potentials from traveling back toward the cell body. Therefore, an action potential that starts at the axon hillock moves in only one direction, which is toward the synaptic terminals. This can also be seen in the figure. In part 1, an action potential is generated as Na plus flows inward across the membrane at one location. In the second part, the depolarization of the action potential spreads to the neighboring region of the membrane, reinitiating the action potential there. To the left of this region, the membrane is repolarized as K plus flows outward. The depolarization-repolarization process is repeated in the next region of the membrane. In this way, local currents of ions across the plasma membrane cause the action potential to be promoted along the length of the axon.
Vertebrate axons are enveloped with these layers of insulated fatty tissues that increase the speed of the action potential. These are called myelin sheaths. The insulation provided by the myelin sheaths allow the depolarization that occurs during the action potential to spread farther inside the axon, and this allows further parts of the axon to reach the threshold faster. The parts of the axon between the myelin sheaths are called the nodes or ranvier. At these nodes, only do the extracellular fluid come in contact with the axon. The voltage-gated sodium channels are also only present in the nodes. Therefore, the action potential only occurs in these regions. These adaptations allow for saltatory conduction, which is when the action potential seems to jump from node to node across the axon. So what happens is the current produced during the rising action of the action potential travels from one node to the other, where the membrane is depolarized and the action potential is triggered again.
Lastly, we're going to take a look at how this signal is transmitted from neuron to neuron.
Neurons transfer information through synapses. There are two types: electrical synapses, in which there is a gap junction through which a current travels from neuron to neuron, and the more common type, which are chemical synapses, in which neurotransmitters are released from the presynaptic neuron to the postsynaptic neuron. These neurotransmitters are held in multi-membrane synaptic vesicles in the presynaptic terminal. When the action potential reaches the synaptic terminal, it depolarizes the membrane, which in turn opens the voltage-gated calcium ion channels. The increased concentration of calcium ions in the terminal causes the synaptic vesicles to release the neurotransmitters. These neurotransmitters then diffuse across the space between the presynaptic and postsynaptic neuron. This space is called the synaptic cleft. The neurotransmitters then binds to the ligand-gated ion channels in the postsynaptic membrane and allow for ion transfer of sodium and potassium ions. Inhibitory neurotransmitters like GABA hyperpolarize the membrane, while excitatory neurotransmitters like glutamate depolarize the membrane, which leads to a higher chance of another action potential.